Now, besides red blood cells and white blood cells, the other foreign elements in the blood are called platelets. Now, platelets aren't really cells, but they do come from a type of cell called a megakaryocyte. Megakaryocytes are in the bone marrow, and what happens is, parts of their cytoplasm form vesicles and branch off. So this is why platelets are really not considered cells, but they're just pieces of a larger cell. And platelets' function is basically to clot blood, but more than just clotting blood, it does produce growth factors. Growth factors are going to aid in repair of the blood vessels, and it's also going to release chemicals that are called vasoconstrictors. They're going to enhance the constriction of the blood vessels once an injury does occur. Later on, they will form temporary platelet plugs here, right before the blood fully clots. And at the end here, they will dissolve the old clots, as well as phagocytized bacteria. So they're part of our immune response as well. And finally, they'll also attract other white blood cells to the site of an infection. So wherever there's a swelling, a damage to the vessels, more white blood cells will be drawn to this area by their actions. The control of bleeding is called hemostasis, and basically hemostasis involves the platelets, and basically involves 3 steps. The first step in hemostasis is called a vascular spasm. What happens is the blood vessels will constrict. Now, the constriction is going to cause less blood to be lost, because the blood vessels are being made smaller. Now, as I put up here on the slide here, direct injury to a smooth muscle and the pain receptors innervated are going to actually cause the blood vessels to constrict and try to squeeze themselves off. This is just a really good measure here to kind of provide time for the other mechanisms that we're going to follow here to take place. Step 2, we get the platelet plug formation. Now, once the blood vessel is broken, that nice smooth endothelial lining is damaged, collagen fibers are going to be exposed. And once those collagen fibers are exposed and they start sticking to the platelets become spike shaped, and they become very sticky, and they start blocking off the opening. Now they, as I put up here, they degranulate, which means that they start releasing all kinds of new substances. Again, those are the substances that are going to attract more white blood cells and attract more platelets and provide the feedback here, or what I call up here positive feedback cycle. When you get more of the platelets breaking off here, you get a more clotting. So the one event leads to an increase in the next, and just keeps getting larger and larger, stronger and stronger effect until the blood vessel is completely closed off. Third step, hemostasis, this is the actual coagulation. Okay, coagulation. What happens is the fibrin in the blood, which is the proteins, becomes fibrinogen, and this is going to completely close off and help to transform the blood from a liquid to more of a gel. And generally, as you've seen clots before here, the blood completely clots off here. There's no more blood leaving the tissues. Okay, so to sum up here what's happening, first of all, if there's a blood vessel damaged, the blood vessels will constrict. Try to make the opening smaller here, but as it does this here, it's going to expose collagen fibers, which are going to cause the platelets to adhere to them, and the more platelets that adhere, eventually it's going to plug up very quickly. And then final step, fibrin fibers are going to become fibrinogen, and it's going to completely form a network here to kind of seal off this opening completely. Now, it's important to remember that these 3 steps occur very quickly. Now, what's going to stop the blood from clotting anytime it wants? Well, there are several mechanisms that the body's evolved with here to prevent coagulation and clotting from occurring just any place it wants. One is platelet repulsion, which basically means that platelets are naturally going to platelets will not adhere to each other's coating unless there's been some some stimulus, for example, running into a collagen fiber from a broken blood vessel. So platelets just normally repel each other. Second, there's not very many platelets in the blood altogether, so the fact that they're diluted, less chance they're going to run into each other. And normally, rapidly flowing blood is going to help dilute the thrombin, part of the platelets here. So just as long as you've got blood flowing, everything's going to keep the blood from clotting. That's why people in hospitals, tend to get people moving as much as they can, and why bedridden patients, do suffer from clots. And our bodies naturally produce some anticoagulants, antithrombin for one, and heparin. These are two natural anticoagulants. Antithrombin is produced by the liver, and what antithrombin does is it deactivates thrombin before it can act on the fibrinogen to actually form the clot. Heparin is secreted by the basophils and mast cells, and what that does is it interferes with the formation of a prothrombin activator, so just going to stop the clot from ever forming. Some coagulation disorders that you may have heard about, basically unwanted coagulation. The term embolism, that's an unwanted clot that's traveling in a blood vessel. So they could form someplace else and travel to another part of your body. Generally they'll form like in the legs, and they may travel to the heart or the lungs. Thrombosis, this is abnormal clotting in an unbroken vessel. And again, this is going to often occur most often in the legs because the blood vessels there, especially if they're inactive, bedridden or whatever, you're going to get low blood flow, and the thrombosis is going to likely form. And it'll probably break off and probably cause death. And one of those things here is infarction or tissue death. Okay, infarction or tissue death occurs if the blood clots is supplied to an organ. It might be a myocardial infarction, like in the heart, or it could be any other organ here, which could result in a stroke, for example, if it occurs in the brain. Quite a few people die. I won't tell you the number, but somewhere around 650,000 people a year die from some sort of tissue damage like this. Now, too little clotting, hemophilia. Hemophilia is a sex-linked recessive trait, so it's going to show up in men, in men primarily. It'd be very rare if it ever occurs in a woman because, it's going to be sex-linked recessive. And what happens with this here is the blood will not clot. So physical exertion might cause bleeding and a lot of pain. And in hemophilia, also called the bleeder's disease, your blood just doesn't want to clot because it's lacking one or more clotting factors. Now, clotting of blood cells has been a standard practice here, observing the way blood clots in different kinds of, with antibodies and everything, antigens. Has been the basis for blood typing. That's what we're going to get into right here. Red blood cells have a coating around on the cell membrane. It is a cell, remember. So there are going to be proteins and glycolipids on the surface here that are going to be unique to the individual. And they can be affected by antibodies. In my picture here, the antibodies are green, and these antibodies are sticking to a red blood cell. And what's going to happen is it's going to cause all these red blood cells to clump together, and that's the basis that we use here for determining blood types. There are 4 blood types in humans: type O, type A, type B, and type AB. Now, the main thing to remember is this. If you have let's say type A blood, you have this carbohydrate chain attached to your surface of your red blood cells. If you have type B, you have a different type. By the colors here, you can tell they're just different. If you have type AB, you have a combination of both. So if you have this antigen, that's what this is, here's an antigen on your red blood cell, and it encounters an antibody, the antibody will attack this antigen. So that's the basis for blood typing. If, for example, you have type A blood, all they did here was they put a drop of blood here and here. They added the antiserum. This has the antibodies for anti-A. So these are anti-A antibodies. They will attack the A antigen. So what happened here was they dropped the antiserum on it. The anti-A antibodies clumped together, caused the clumping here of the A antigens. Now, because there's no B antigens, this is not in the type A blood, the blood remains clear. And you could guess the same thing would happen if type B blood, will not clump in anti-A because, again, it's only against the A. But it will clump in the B because it is against the B. If you have both of these antigens on your red blood cells, type AB, your blood would clump in both. And if you have type O, which is totally different from A or B, the antiserums clump in neither. So what happens if you get a mismatched blood type? In my example here, you've got the example here where a person is receiving a blood donation from somebody who is type A, but this person is type B blood. So as their red blood cells flow in, their blood flows into this vein of the recipient, this type B person is carrying anti-A antibodies. So as soon as those blood cells come in, the anti-A antibodies are going to attack the A antigens on the red blood cells. And that's going to cause these blood cells to start to clump together. And if they block a blood vessel, there's where the problem is going to come in. You're going to restrict the flow of blood to that particular organ. So always remember, if a person is type A blood, they are going to have anti-B antibodies. So I guess you could also say that this person's type A blood coming in here is also got the anti-A antibodies, or excuse me, the anti-B antibodies. So the anti-B antibodies are also going to attack the recipient's blood as well. So both those things together are going to cause these clots to form. So again, type A blood has type B antibodies. Type B blood has anti-A antibodies. Now, could there be a problem here with a Rh negative mother versus an Rh positive baby? Well, this is called the hemolytic disease of newborns. And what happens is this, and we'll discuss this again with more of our immune system. Another blood factor is called the Rh factor. It's another antigen that's on your red blood cells. You either have it or you don't. If an Rh negative mother, her red blood cells do not have that antigen. But an Rh positive baby, its red blood cells have that antigen. So what happens is this. If the baby is Rh positive in the mother, and somehow or other, that baby's blood becomes mixed with the mother's, okay? The mother is going to see those Rh positive blood cells as a foreign invader. So the mother's body between pregnancies starts forming anti-D or anti-Rh, antibodies floating around in her bloodstream. With a second pregnancy, those antibodies can be transferred through the placenta and absorbed by the baby. And now those antibodies are going to view the baby's blood as a foreign invader, and they're going to start, destroying those red blood cells. Could result in serious complications, but from what I understand, they generally have a chance to do a transfusion, so the baby can survive. Most of the antibodies really don't develop completely, for the first few months after birth. So anymore, what they're doing to the mothers is, if there is a question about it, they give her a shot to desynthesize her, so that she doesn't, form the anti-Rh antibodies or the anti-D agglutinins, between pregnancies. So her second pregnancy would be perfectly normal, just like the first.